Hello, my name is Kunjumon Vadakkan. I study the nervous system to understand how it operates. I have a conflict of interest to declare. We are still working hard to understand how the brain operates. Without having a clear-cut knowledge of that operational mechanism, it is difficult to understand the disorders of the system, such as neurodegenerative and psychiatric disorders. But what is encouraging is that we have a list of large number of findings from both normal functions of the system and defects of the system, such as loss of function and gain of function states seen in various neurodegenerative and psychiatric disorders. So if we can put together the findings from various levels to synthesize a hypothesis for the operation of the nervous system, then we should be able to go and examine whether the defects of that system can explain various loss and gain of function states seen in the neurodegenerative and psychiatric disorders. In a previous presentation, we have seen the list of all the normal findings from various levels, and we have seen the derivation of a hypothesis for the nervous system functions. In this presentation, we will see a list of common symptoms and signs of the defects of the system. Number one, 80% of the neurodegenerative disorders are sporadic in nature. 80% are sporadic in nature. Sporadic in nature. That means that it is possible that there could be a combination of different factors that can cause the neurodegenerative disorders. The second feature is contiguous lateral spread of the pathology. See, the neuronal orders of the cortex, we will see the neuronal orders in this way. This is neuronal order 1 and 2, and it keeps going like that. Neurodegenerative disorders spread laterally in most of the disorders. So that lateral spread of degenerative disease cannot be explained by synaptic transmission defects alone. So we have to find out a reason why there is contiguous lateral spread. The third feature is presence of abnormal proteins. And we have to find out a reason why abnormal proteins are getting expressed. And these proteins also form aggregates. And another feature is heterogeneity. Heterogeneity of abnormal proteins. This means that the abnormal proteins that are getting expressed vary from region to region within the nervous system of a single patient. And also, these abnormally expressed proteins are different in different patients. So that means there are no two patients having the same pattern of abnormal protein expression in neurodegenerative disorders. So we have to go and find out a clear-cut reason from the hypothesized normal functions to understand the reason for such a heterogeneity. These disorders also produce heterogeneity of clinical features. The common clinical features of these disorders include hallucinations, cognitive defects, seizures, then reduced attention, and change in consciousness, change in conscious states. So these are common clinical features, and therefore we should be able to look at the normal mechanism of their formation from the hypothesized mechanism to see why these features are formed in neurodegenerative and psychiatric disorders as a result of defects in the normal mechanism. Another feature is the effect of dopamine. Dopamine is known to promote learning, and the same dopamine is used to improve the symptoms of the hypokinetic movement disorders seen in neurodegenerative disorders, movement disorders. And also dopamine blockers help to reduce hallucinations. And the next feature is that large number of viral fusion proteins cause neurodegenerative disorders. For example, HSV virus infection, enveloped viruses such as lentivirus have fusion proteins. An example is Zika virus. They have fusion proteins, and infection with these viruses can lead to neurodegenerative changes. So we have to again go and examine the normal mechanism that is hypothesized to see how that can be explained. Another feature is aging is contributing to neurodegenerative changes. Also, defects in essential fatty acids in diet, essential fatty acids in the diet also is implicated in contributing to neurodegenerative changes. Another factor is that anesthetics and certain chemicals can lead to neurodegeneration. In this regard, we have to also look and see how dopamine reduce the threshold for anesthetics. So now we have an advantage in that we can go and examine the hypothesized normal mechanism and see if defects of that mechanism can lead to all these changes. So let us see the normal mechanism. So this will be a repetition of some of the previous presentations that explains normal operation of the mechanism. So we have seen that. So this is the presynaptic terminal, and this is the postsynaptic terminal. These are lipid bilayers. So this is pre, this is post, and based on the semblance hypothesis, there will be formation of a functional link between these two postsynaptic membranes during associative learning. And during memory retrieval, one of the cue stimuli arrive through this synapse, depolarize this postsynaptic terminal, and reactivate this interpostsynaptic functional link, and then the depolarization spread to this postsynaptic terminal. Since this postsynaptic terminal is receiving condral release of neurotransmitter molecules all the time from this presynaptic terminal, according to semblance hypothesis, any arrival of depolarization by a lateral route towards this interlinked spine will be able to trick this spine to hallucinate that it is receiving sensory stimuli from the sensory receptors that is coming to this presynaptic terminal. So that can explain units of virtual inner sensations, and that can explain how the first person inner sensations of various higher brain functions such as consciousness, perception, memory, thought process, and imagination, all those can be explained. Now, the formation of these interpostsynaptic functional links are of different types. The number one is hydration exclusion. Hydration exclusion. That means when we try to study the mechanism of interaction between the lipid membranes using artificial lipid bilayers, we have observed that when those two lipid membranes are brought very close, maybe to closer by 15 to 20 Armstrongs, then they will get repelled from each other by water hydration that is present there. So normally water hydration is present, and exclusion of that water hydration will give rise to an interpostsynaptic functional link. A very high energy is required. Experiments have shown that 100 atmosphere pressure is required to exclude the water hydration. Calculations have shown that it requires around 50 kilocalories per mole of energy for excluding that water hydration. So even by removal of water of hydration, the interaction of the lipid bilayers can take place only at an area of 10 nanometer squared. So that is a very small area, but that is enough for an interpostsynaptic functional link to form, through which the depolarization can spread from one postsynaptic terminal to the interlinked postsynaptic terminal. Now, in conditions where we have dopaminergic inputs or dopaminergic synapses to this postsynaptic terminal, the dopamine will cause enlargement of the dendritic spine. So conditions that lead to enlargement of dendritic spine now can cause the interpostsynaptic functional link to form and advance further to form hemifusion. So now instead of 4 layers of membranes here, we have only 2 layers of membranes. So this process is called hemifusion. Hemifusion. So this is a reversible process. It's a highly reversible process. Once the factors are removed, that means once the swelling of the spine is gone away, the hemifusion will reverse back to the independent membrane state. But other factors such as changes in membrane composition, changes in lipid composition, or reduced sodium in the extracellular matrix space will reduce the water hydration in the extracellular matrix space and will allow the dendritic spine to hemifuse. But once the conditions reverse back, the hemifusion will reverse back to the independent membrane state. But the next stage is that hemifusion is an intermediate stage towards fusion. So this is seen in biological systems. Say for example, at the presynaptic terminals, membrane vesicles are fusing with the presynaptic terminal membrane, and it causes release of the contents of the vesicles into the synaptic cleft. That process is a fusion process. So at the presynaptic terminal, this fusion process is taking place all the time. According to semblance hypothesis, it is advantageous for the system to stop the changes at the level of the hemifusion. And we have also told that if these hemifused membranes can be stabilized for a long period of time, then that can lead to the induction of units of inner sensations for a long period of time, explaining long-term memory. We expect that the machineries that cause membrane fusion to present at the postsynaptic terminal as well, but it is expected that there will be some checkpoint mechanisms to prevent fusion. So to prevent fusion. So let us see some of the key molecules that are important in bringing about certain changes at the postsynaptic membrane level. The first protein is Snare proteins. These proteins are seen at the presynaptic terminal that are taking part in the membrane vesicle fusion. And these snare proteins can lead to both hemifusion and fusion. So vesicle studies have shown that both hemifusion and fusion are the end products of the action of snare proteins. But we expect to have a checkpoint mechanism to prevent fusion at the postsynaptic membrane level by the snare proteins. Snare proteins also form an alpha helical bundle that bring the membranes together. And also self-assembly of the snare proteins can also again bring the membranes together and can lead to fusion changes. But again, we expect a checkpoint mechanism to prevent fusion from the hemifused state. And another factor is that the activity of the snare proteins depends on the lipid membrane composition. Lipid membrane composition. Also, snare proteins are essential for LTP, long-term potentiation. The latter can be explained in terms of interpostsynaptic functional link formation. Another protein is of mention is AMPA receptor subunits. AMPA receptor subunits, namely GLUR1 at the glutamatergic synapses. And the GLUR1 is present at the postsynaptic terminal within membrane vesicles. So again, these are lipid bilayers like the postsynaptic and presynaptic membranes. So the GLUR1 containing vesicle moves towards the lateral aspect of this postsynaptic membrane and fuse there and release the GLUR1 subunit to this postsynaptic membrane, which in turn forms functional AMPA receptor. So at this point we expect some kind of a fusion process that enables the release of the GLUR1 subunit towards the membrane. Studies have shown that AMPA receptors are present at around 25 nanometers away from the synaptic margin. So that location where AMPA receptors are observed, if this vesicle exocytosis is taking place at that location, it can lead to membrane reorganization, which will help the hemifusion process. So that is a candidate mechanism that can help the formation of interpostsynaptic functional link, that is the membrane hemifusion stage. Now let us see what happens if the postsynaptic membrane undergoes the fusion state. If the checkpoint mechanism of the snare protein fusion that prevents hemifusion to fusion is not present, then it will lead to fusion. What is going to happen when there is fusion between the postsynaptic membranes? Now, if you take a cross section at this point and if you put it here, we will get pictures like this. This occurs at the time of associative learning according to semblance hypothesis, and continuous learning will cause formation of further interpostsynaptic functional links, and these are going to form a group. And here we have a different group of interpostsynaptic functional links. Now, if there is excessive fusion changes that occur secondary to defects in the lipid membranes, or due to viral fusion proteins, or due to reduced sodium at the extracellular matrix space, that will lead to excessive hemifusion and fusion, and it is likely that these group of interlinked spines, they have to be maintained separate for their function, but excessive hemiffusion and fusion will cause a functional link to form between these two islets, and that is highly deleterious because they are not associated together. They are completely different items that were associated together, and for the specificity of the retrieved memories, they have to remain isolated. If they get connected, that will lead to reduced memory, because we have already seen that memories, the units of inner sensations, are formed by tricking this interlinked postsynaptic terminal by the cue stimulus to hallucinate that the sensory receptors are getting activated here, which are contributing to the inner sensation of the memory of the retrieved item. So, in order to maintain specificity of the retrieved memories, we have to keep the islet of interlinked spines separate. If we mix them together, it will cause reduced specificity of the retrieved memories, and that can explain cognitive defects. So, we explained how cognitive defects can be formed. Now, it can also lead to internal sensation of a completely unassociatively learned sensory stimuli. So, for example, when the cue stimulus arrives to one of the interlinked spines, it is only supposed to traverse through these interlinked spines, inducing virtual inner sensations of previously associated items. But if it goes to a completely unassociated islet of interlinked spines, then it can lead to hallucinations. And also a rapid spread of this depolarization across different islets of interlinked spines can explain seizures, which I will be explaining in a separate presentation. Now, since we are going to explain the framework for consciousness that depends on the reactivation of interlinked spines in a background state during oscillating extracellular potentials, which will induce units of inner sensations at large number of interlinked spines that contribute to a net semblance, and that was hypothesized as a framework for consciousness. So, formation of any abnormal links between unassociated islets of interlinked spines can cause reduced conscious state. So, during aging, if the lipid membrane composition is getting changed due to defects in the lipid synthesis, elongation or desaturation process, that can lead to changes in the composition of the membranes, and that may favor fusion changes. Viral fusion proteins seen in large number of viral infections can directly cause membrane fusion, and dopamine is causing enlargement of the dendritic spine that favor membrane hemifusion if it is at an appropriate concentration, but any excessive dopamine can lead to fusion changes. Now, essential fatty acid is important in maintaining a normal lipid membrane composition. Anesthetics. In a separate presentation, I will explain a potential mechanism of action of anesthetic, because anesthetics cause loss of consciousness. Formation of any non-specific interpostsynaptic functional link will change the background semblance during normal background oscillatory potentials. So, any changes that caused formation of non-specific functional links can lead to the loss of consciousness, and that can explain why dopamine is reducing the threshold for anesthetics, because dopamine is going to cause enlargement of the spines, which can explain the formation of non-specific functional links, which is the mechanism of action of anesthetics. So, therefore, dopamine is causing reduced threshold for anesthetics, and why there is 80% are sporadic in nature. From this explanation, we can see that there are large number of factors that are associated to cause membrane fusion, such as presence of dopamine, aging-induced changes in lipid composition, reduced sodium in the extracellular matrix space, viral fusion proteins, and the anesthetics. So large number of features should come together to cause membrane fusion, which is responsible for the neurodegenerative changes. Now, why there is contiguous lateral spread? Because if you look at here, the spread of pathology is taking place through the interpostsynaptic function link. So the spread is occurring in this direction. So that can explain why there is spread of pathology from cell to cell within one neuronal order. So now we have to explain the expression of abnormal proteins and their aggregation. If you look very carefully here, by the formation of this fusion, the cell contents mix together. Experiments have clearly shown that if this is one neuron, and if this is its dendritic tree, and this is a spine, and if it forms a fusion there with the neighboring spine of a neighboring neuron, this is neuron 1, neuron 2. So these are adjacent neurons within the same neuronal order. Studies have shown that the protein expression that are present within the cytoplasm of two adjacent neurons within a same neuronal order is different. Therefore, if we allow the contents of these dendritic spines to mix with each other, there will be a cell reaction towards the abnormal proteins that are arriving to its cytoplasm. So normally what we expect is that initially there will be expression of abnormal protein as a result, because a foreign protein is arriving at this dendritic spine, and therefore the cell body is going to react to that abnormal protein by expressing certain proteins which are normal to this neuron. And those proteins are likely to get aggregated because there is continuous cell to cell interconnection, there is cytoplasmic content mixing that can lead to protein aggregation. And then we can say there is heterogeneity of abnormal proteins. Since the protein expressed at the neighboring neurons within a neuronal order is completely different, therefore this neuron is going to produce certain abnormal protein which will be different from the abnormal protein that are expressed by this neuron. So as we move from neuron to neuron within a neuronal order, the abnormal proteins that are getting expressed will change. So that can explain the heterogeneity of abnormal protein and therefore the clinical features. Another feature that we have to explain is how will the interpostsynaptic membrane fusion be responded by these two neurons. Initially, it is expected that there will be mechanisms that will enable closure of that fused area. So that could be by plugging that fused area with some proteins or some other mechanisms. So that will be stage number 1. And if the cells cannot block that fused area, then the cells will start cellular response mechanisms whereby this neuron might detach that spine at the spine neck level, and that will cause the loss of dendritic spine. Loss of dendritic spines is a common feature that is seen in most of the neurodegenerative disorders. The next mechanism will be that if the cells cannot block this fused area, and if the cells cannot cause a detachment or the separation of that spine at the spine neck level, then it is advantageous for this neuron to die. So autophagic mechanisms will be triggered for the death of this neuron. So that will enable this neuron to survive. So neuronal loss is also a feature seen in many neurodegenerative disorders. In summary, based on the semblance hypothesis, there is formation of interpostsynaptic functional links. Large number of changes of this mechanism can lead to interpostsynaptic fusion. Any fusion can cause cytoplasmic content mixing. Any cytoplasmic content mixing can cause expression of abnormal protein, their aggregation, and can explain various features here. From the defects of the formation of units of inner sensations, we can explain large number of clinical features such as hallucinations, cognitive defects, seizures, changes in conscious states, etc. And all other features can be explained as defects in the normal mechanism as hypothesized by semblance hypothesis. I sincerely hope that you understood the logic behind this presentation. We should examine this hypothesis and these loss of function states seen in various neurodegenerative and psychiatric disorders. Thank you very much for your attention and have a great day.